Okay, so I'm now going to follow up on that, a really interesting conversation in the presentation we've just seen from Justin. And actually I'm going to focus a lot more on the formulation attributes and how we can use that to improve delivery and targeting. So it's really about the nanoparticles that we've been hearing. So nanoparticles, you know there's plenty of them. We've just heard all the nice work that Justin's been doing with the lipid nanoparticles and mRNA. But they also cover, obviously, liposomes, polymeric nanoparticles. We can go into solid lipid nanoparticles, emulsions, and the list goes on, cubosomes, ISCOMs, various light particles. There's many options for us to consider in nanoparticles. And really, their use as a vaccine adjuvant is not that new. The first paper where it cites nanoparticle and vaccine was back in 1981, where they're looking at an influenza-type vaccine. And if you go more general, looking at the use of particulate systems, you can go back to 1976, where the first paper looked at using liposomes by Gregory Addis, who was looking at using liposomes as an adjuvant system. So certainly not a new idea, but our understanding of how to improve them and to exploit them is certainly continuing to grow. And you can see at the moment we've got about 2,800 papers on PubMed when you do a search for nanoparticles and vaccines. So the role of nanoparticles is really kind of twofold. Firstly, we're looking at delivery. That's the key thing. We want to get our antigen to the site of action, have it protected and have it delivered. And then when it gets there, depending on what we're delivering, if it's mRNA or a protein or a subunit, we might want to activate the cell. So we need to stimulate or have an immune response, potentially an antigen presenting cell. So we have this dual aspect coming on. And really a lot of what is going on is driven by the antigen-adjuvant interaction or the antigen nanoparticle. Now a lot of the very early work look at just mixing nanoparticles with an antigen. But over time we've obviously changed that. We're now looking at absorption to the nanoparticles. We can encapsulate within the nanoparticles, or we can even conjugate onto the surface. So there's many options we can consider in this aspect. And two of the most commonly considered, I would say, are the absorption, particularly now as we move more into the cationic systems or the ionizable with the subunits or with the mRNA type systems, but also encapsulating, putting something inside, protecting it, and then allowing delivery. Obviously absorption, if you've got the electrostatic, allows us to have higher interactions, higher payloads generally. However, if we go with encapsulation, we're not relying on an electrostatic, so you can have a bit more functionality and options over your formulation in that respect, but you tend to get slightly lower antigen loading. So if we look at encapsulation first, so this is us trying to put an antigen within a nanoparticle. We do have to consider what charge we want. Do we want it positive, negative, or neutral? The size of the particle is important. Although we talk about nanoparticles, there has been a lot of work on microspheres and larger particles. How we make the composition, do we make them non-immunogenic or do we make them immunostimulatory? And we do have to consider manufacture. Can we actually make these cheaply, easily, so that they could form a global vaccine campaign? And in terms of charge, size and composition, they all have a direct impact on how we deliver the antigen and how immunostimulatory we are looking at. So to look at how the adjuvant charge dictates distribution of an antigen, we first started looking at formulations of liposomes with different charges. So here we have 3 liposome systems, distereophosphatidylcholine with cholesterol, so it's a neutral liposome. We made them about 100 nanometers in size. We then added in a negatively charged lipid, phosphatidylserine, to give us a liposome with a highly negative charge, with about 66 millivolts negative. And then we also made cationic ones. So we added in DOTAP, a well-known cationic lipid. Again, the liposomes are about 100 nanometers in size, but very positively charged this time. And we injected these intramuscularly, and we follow the fate of them to see where they go. So what you can see here is the X's show that the cationic liposomes are sitting at the injection site. So we inject them in, they stay at the injection site due to the cationic nature. The black triangles are our neutral liposomes. You see them flowing away from the injection site. And our open circles are our negatively charged liposomes, again flowing away from the injection site. And you can see as they flow away, they're more likely to appear at the lymph node. So you can see our negatively charged liposomes flowing away quickly, arriving at the draining lymph node, whereas our cationic ones are sitting at that injection site, can't move, so remain there. So you can see the role of charge very easily. The adjuvant charge is dictating the distribution. And why is that? Well, basically the charge is due to biological interactions. If you've got something very cationic, when you mix it with protein, as can be seen here, the particle size really grows. So something neutral won't interact with the biological environment. They stay as individual liposomes. They can move away freely. Something cationic, the positive liposomes, when you mix them with any sort of protein at high levels, you get this aggregation, clumping, and you actually form the depot at the injection site. So you can see instantly charge is a very important factor and a basic physical function of our formulation. Now, particle size does have a place. It depends on the charge of your formulation. So if you're working with neutral or negatively charged liposomes, size will be an important factor in the flow from the particle from the injection site. So here we made 2 liposome formulations, both negatively charged. Our upper one you can see is about 900 nanometers, or we have very small SUVs around 75 nanometers. So you can see the larger ones, shown by the black square in the top graph, are injected in the injection site, take a while to move compared to our open circles, which are our small unilamellar vesicles. They're flowing away. And again, if you look at the bottom graph, you can see they're flowing away from the injection site and arriving at the lymph node, whereas the larger liposomes are still stuck at the lymph at the injection site and probably take longer to get there. So both size and charge are important factors we consider. But also, how do they interact with the cells? And that was kind of what I was just asking Justin about. How do they get taken up? So we were tracking them using fluorescent liposomes, so fluorescently labeled liposomes. We'll look at uptake into the THP-derived macrophages. And this is what we have ongoing with Andrew Devitt down at Aston University in Birmingham. So what we can see is definitely the smaller liposomes, you can see in the open circles again, are easily taken up by the macrophages. So they get taken up, and we measure this by fluorescence uptake compared to our liposomes. And we can monitor that either in the total number of liposomes taken up or lipid content. And in both cases, we can see the macrophages are easily more likely to take up the smaller ones. So whether the movement from the injection site is just physical or the cells help us is a question we're still looking into. So we can see that. So size and charge important. So what about putting the antigen inside? How do we get it encapsulated? We know what dictates delivery. How do we get the antigen inside? So this is where we've been working a lot with microfluidic systems. So we've been working with this now for probably about 10 to 15 years now, looking at that. So basically we have our lipid in the solvent phase. We put our protein, our subunit antigen in the aqueous phase. We send it through the microfluidics, through the herringbone mixer, and we produce liposomes. So the full setup, we've been really thinking about how this scales and how you would implement this as a manufacturing. So we have our lipids in methanol or ethanol. We have our protein in PBS. They go through the microfluidics in the process step. We're using the nanoassembler for that. And the in-process controls we look at are basically particle size, as you would imagine. PDI and the dispersity, important for us in terms of ability to filter and quantify the product. Zeta potential, obviously very important. We saw how charge impacts on where they go. And also lipid recovery. We don't want to lose all our lipid within the microfluidic cartridge. So we then purify to remove any non-entrapped protein and also any residual solvents. So we've been using cross-flow filtration. Again, we use a small-scale spectrum, but it's, again, completely scalable process. And again, here we're quantifying to make sure we get rid of solvent, remove free protein, we keep our liposomes, they're not lost in the system, and our adjuvant retains its attributes in terms of size, charge, etc. So those are all things we monitor, and we have all that system in place now, and we are able to show we get the same product out. And I've previously presented that, so I won't go into that too much. And basically, once you've got that, you've got your product that you can basically sign off under QC. So that's the whole process train we've worked up, and it works nicely in a scalable format. And we've been using microfluidics mainly because of its really high loading compared to other systems. So here we have a graph of protein encapsulation on one side, and we've got size. So microfluidics, we're getting about 35 to 40% protein loading. Particle size around 70 to 80 nanometers. Whereas sonication, the particle size is about 200 nanometers. Now I admit we could get that smaller, but really there was no point sonicating more. Our protein loading was very small already, around about 6%, so we could, if we take the size down any smaller, then the loading's going to down even more. Extrusion was another option for us. Again, you can take down a particle size very easily with extrusion. We got the particles down to about 150 nanometers. There was no point going further because already our entrapment was down to 5%. So you can see the advantage of microfluidics giving us very small liposomes with very nice loading, nice and high, and that's because it's a single step bottom-up process where you're bringing it all together and not having to add a size reduction step. And in terms of the protein stability, so here we did some circular dichromism. So you're looking at ova native versus after it's been put through microfluidics, and we see the same structural spectrum. So the ova is nice and happy going through the microfluidics cartridge, which is always a concern. So we are able to encapsulate the protein without degradation or loss of conformation. So when we look at microfluidics, we've obviously various parameters. We've got the ratio we mix the solvent to the aqueous phase, so that's flow rate ratio we would refer that to. And then we've got how quickly we run the system. So the current nanoassembler can run up to 20 mils, but obviously per minute the larger systems run faster. And the things we wanted to consider was the concentration of lipid, how we make the liposomes, and those two parameters. And things we were considering, obviously we saw the importance of size and charge, so we were keeping that. We need to make sure we got our lipids back out. We need to make sure our protein was loaded, and also we wanted to check if these systems worked in vivo. So in terms of, these are basic liposome formulations, so we've got phosphatidylcholine and cholesterol, everybody's kind of starter product we tend to look at. And then we've increased the phospholipid chain length from phosphatidylcholine up to distereophosphatidylcholine, and we've increased the concentration as well of initial lipid. And we see as the carbon chain length gets a bit longer, we do drive down the liposome size slightly from about 100 nanometers down to about 50 nanometers for DSPC. The lipid concentration you start with has an important parameter. If you work with very low concentrations, you tend to get slightly larger particles, more heterogeneous. But once you get over roughly about 4 mg per mil initial lipid concentration, you flatline out and you get very reproducible products, no matter how high you go in concentration. And throughout this, the PDI is always very low, below 0.2, which we take as a gauge of a nice reproducible product that will be easy to format and characterize. So in terms of temperature, so a lot of us, if we've been training in the liposome field for many years, we're always told that we had to work above the transition temperature, particularly with long chain lipids like distereophosphatidylcholine, and you needed to work above the transition so that you could form liposomes. Indeed, many of the products on the market with high transition involve a heating stage at 60 degrees. So we wondered if we were going from a bottom-up process, did we actually need to involve this temperature step, and if we could avoid it? The answer is basically yes. With the microfluidic system, you can see here we have a graph where we've formed liposomes at temperatures from room temperature up to 60. And for example, if you look at the green line, that's a weight ratio of 10 to 5 or 2 to 1 DSPC to cholesterol. So if you were doing this in round bottom flask, convention would say you would heat up the lipid to hydrate it, but here we were doing it at room temperature with no impact on size. So that was really good. We've taken out a process step within the manufacture, so we can easily produce a range of liposomes with different cholesterol concentrations at room temperature, which is great to be able to achieve. And again, our PDI, nice and low, below 0.2. So we know we can get rid of temperature. We know what kind of concentrations we work at. What about the mixing ratio of lipid to aqueous phase? So you've got the 1:1, 3:1, 5:1 ratios we looked at, and generally we find as you increase the ratio, you reduce particle size. And as always, PDI was nicely below 0.2. We tend to now select a 3:1 ratio because it always gives us a nice low size, reproducible, but we're not too dilute a product. But you can obviously, as you can see here, modify your particle size by selecting whatever flow rate works best for you. So, in terms of particle size, we've got that thing. What about protein loading? So again, here we have 4 formulations where we've increased the phospholipid chain length from PC to DSPC. And you can see we get nice, similar protein loading, 35 to 40%, irrespective of what phospholipid we use. So we're seeing basically here the protein loading is process controlled and not driven by the lipid. And actually, if you plot the graph of log encapsulation versus log lipid, you're really looking to see if there's a correlation here. And if there was, then we would say that it's more driven by the phospholipid. But actually, you can see here that R values are quite low and the gradient's quite low. So it's actually driven by the production system and not our lipid concentration, which is good to know. So, as I say, we looked at flow rate ratio. So this is now antigen loading. So we can see this 3:1, 5:1, it controls size and it does control protein loading. So the 3:1 giving us good loading, 5:1 reducing it down a bit. In terms of the speed we run the system, generally above 10 mils per minute gives us reproducible data. 5 for us is a bit slow and it's not giving us the reproducibility we look. But also it's important to note that the flow rate ratio is giving us different release rates. So when we make liposomes at a 3:1 or a 5:1, we actually see slightly different release patterns. So it's important to actually work with your flow rate ratio, and once you've optimized it, stick with that. We have found it's not interchangeable in terms of release rates. So this is release rate of over from our neutral liposomes. So in terms of anionic liposomes, we looked at them, how easily they can move. Again, very similar story. 3:1 flow rate ratio gives us higher loading. Particle size is less impacted in this case. We can use speeds between 5 and 20 mils per minute, and that's on a single cartridge. Obviously, you can stack up more cartridges. And in terms of encapsulation efficiency, above 10 mils per min is very reproducible. So we've kind of defined the parameters here. We like to work at 3:1, it gives us good data, and we can run at speeds between 10 and 20 mils per minute. On the nanoassembler, obviously, if you scale up to the other systems, it runs faster. So, in terms of concentration, sometimes we might want to concentrate up. Well, we can do that with the cross-flow filtration, TFF. So here we have a graph where we've basically concentrated up 1-fold, 2-fold, or 4-fold. Particles are quite happy, the liposomes. We get nice reproducible product. And in terms of scaling up from the nanoassembler to the larger blaze system that runs basically 10 times scale up, in the system we get very reproducible data, so the particle size and loading is the same. Straight off one, same parameters, no further optimization onto the larger scale. Because it's the same system basically scaled up, so it's really easy to scale up and scale down your production as required. So all that data on loading was with OVA. And you can see here, so what we have here is a couple of liposome formulations. We've got the yellow is when we've entrapped OVA, 23 tells us it's 23%. The white is when we've entrapped tuberculosis subunit antigen, H56. So again, about 20%. But we've also tested a range of other proteins. So again, the yellow you can see is the OVA. We've got bovine serum albumin in there, and we've also put insulin. So it's very reproducible in terms of protein loading and what we can achieve with these formulations. So basically you can see we can encapsulate, we get good loading, it's process controlled, and it's easy to manufacture. So we can do that, and we can make the liposomes in whatever charge we choose. But one thing to note is basically if you have anionic liposomes, they tend not to be as immunogenic. They are more designed for delivery of proteins. So here we did check antibody responses. So we've got the free antigen H56, which is tuberculosis antigen, versus the antigen encapsulated in negatively charged liposomes. So we increase our log titers, but we tend not to increase our interferon gamma. And for tuberculosis vaccines, that's our correlative protection. So we know we can deliver, but we aren't really improving our cytokine responses as we want. So it's more a good use for delivery of proteins in this respect. But one thing we did consider, and again that was back to what we were discussing about, do we need to get this to the lymph node? And if we get it there, do we want to hold it there longer? So we knew these DSPC cholesterol phosphatidylserine liposomes go to the lymph node, we saw that, so we made them small, but how do we get them to stay there, and would that improve it? So we looked at using a Biotin Avidin complex. It had been previously published looking at various other ways. So we're moving from a passive to an active targeting system. So basically you have your liposomes and you put biotin on the surface, you predose with avidin and you form a complex. So we predose with our avidin then moves to the lymph node. We then wait about 30 minutes, we inject our liposomes with the biotin. They then go to the lymph node, we've already seen that. You get a cross-link, and then hopefully they stay at the lymph node and do, is that useful for us? So here we can see the biodistribution again. So these are the liposomes injected. So this is in mouse model, liposomes intramuscular injected. So you can see the top graph is the formulation we saw before, DSPC, cholesterol, phosphatidylserine. So the liposomes with untrapped protein move away rapidly from the injection site as we knew. When we have the biotin added on, so the mice have been pre-dosed with Avidin and then 30 minutes later injected with biotin. The liposomes move away from the injection site at the same rate, so that's good. They're moving away as we would want them to, but do they stay at the lymph node? So here we see the local popliteal lymph nodes. So the top graph is the liposomes as before. They've cleared from the injection site and they've arrived at the lymph node. And then the top ones are then clearing away. But if you look at the bottom graph where we've got biotin and pre-dose with Avidin, the liposomes are going to the lymph node, coming into contact with the Avidin, forming a complex and staying at the lymph node for longer. So we're getting a sustained profile at the draining lymph node. And actually, we also push these liposomes to further lymph nodes outside. So when we don't use a biotin-Avidin complex, we tend not to see the liposomes at the inguinal lymph node. Whereas if we've got this Avidin complex with the biotin, we see these liposomes going further out. So we can now actively target a lymph node should we want to with these anionic formulations. So we can get a protein there or any other drug. So it's really good to see we can manipulate how the liposomes move from the injection site through charge and size, and then we can control how long they stay somewhere else through an additional complex system. So that was all about putting things inside liposomes, but you might not want to do that. We might want to just use surface absorption, and generally this is where our cationic formulations come in. We're normally looking at a cationic liposome electrostatically binding either RNA, DNA, or a subunit antigen, because many of our subunit antigens are negatively charged. Using electrostatics, we get a lovely high load. It's very easy. And the good thing is many cationic lipids can be immunostimulatory. And actually that was where the original gene therapy kind of had some of the problems, because everybody said, oh, cationic lipids are immunogenic. So in this case, it's great because we're using them not only for delivery, but we're actually using them as a stimulatory adjuvant as well. So the formulation we've worked with for many years now in collaboration with Stanton Serum Institute is a combination of two lipids, dimethyl-diammonium bromide and trehalose dibehenate. So it forms a cationic liposome, and we use our weight ratio of 5:1. So the two components, we have the cationic lipid, as I said, DDAB. That allows us to stick the antigen onto the surface, and these liposomes, even without TDB in them, are immunogenic and will give us a modest antibody and Th1 type response. But if we add in the second component to this formulation, the trehalose dibehenate, it can't form liposomes on its own, but when mixed together, you get a very immunogenic liposome which engages the TLR independent Cytokine pathway. So it's like a double approach to that. So these liposomes we had been making for several years using the lipid film hydration method that we all like by Alex Bangham, really easy to use. When we use it, we get liposomes around 400 to 500 nanometers in size. They have a very cationic zeta potential, about 50 millivolts, as we would expect. They have a high transition temperature of around 40 degrees centigrade. So that means they look like a hard particle inside the body. And because they're electrostatically positively charged, we can stick quite a lot of antigen onto the surface. Here we have loading of about 90%. And actually, if you see the fluorescence, we can see the antigen stuck to the surface of the liposomes. So it's a double approach. Now, when we inject these cationic liposomes, so previously I'd shown you negatively charged ones flow away, but the cationics sit at the injection site. So here are these cationic formulation, DDA, TDB. The green line is the liposome, the red line is the antigen. So this is us injecting them intramuscularly into the mouse at the injection site over time. So you see the green line is the liposome sitting forming a depot, holding the antigen. And these formulations give us a high interferon gamma response, which we take as our correlative protection. So we really want a nice, strong response, which we can see here. Now, there are many things we can do to change this formulation. We looked at so many variations. So charge. Obviously, we can take down the cationic charge. As we reduce this positive charge, we help the liposomes flow away from the injection site, and they do reach the lymph node, but equally, that takes down our immune response. I'm not saying they're directly related, but the two things happen. In terms of size with cationics, it really doesn't matter. If we take the size down from large to small, they sit at the injection site the same. We've taken them down from about 500 nanometers to 100 nanometers, and we didn't see that difference in movement, probably because they're aggregating and they're still quite large. It made no difference to how they move, and it made no difference to the immune response. We also looked at fluidity. So I mentioned these are quite hard in nature. Their transition temperature is quite high. So we took in some lipids that brought down the transition temperature, increased fluidity. It helps the liposome move, but again, we saw a reduction in immune response. Other ways to make these liposomes move, we can hide the cationic charge through pegylation, which is well known to mask a charge. That really nicely moves our liposomes away from the injection site to the lymph node. But the presence of the PEG also resulted in a reduced immune response. Finally, we did look at if we put the antigen inside versus the surface, what difference it made. Well, it made no difference to us. So there was no advantage with this particular formulation for us to incorporate it inside. We might as well just stick it onto the surface and make the process as simple as possible. So you can see we've worked through many parameters to optimize it. And actually this cationic charge forming a depot really isn't restricted to liposomes. So here we have liposomes, solid lipid nanoparticles, polymeric nanoparticles in emulsion, and you can see over time from 6 hours to 96 hours. This is the amount of the adjuvant sitting at the injection site. So the blue is sitting at the site of injection, and you can see blue is mainly predominant there. The orange is when they move to popliteal lymph node, the local lymph node. See, this cationic charge drives many of our particles to form a depot and then slowly move to the draining lymph node. So that was great. So we have this brilliant cationic formulation, but as I said, it was made by rotary evaporation, which is not the easiest method to scale up. We know it's not scalable. So we wanted to actually, the reason we originally discussed using microfluidics was to scale this cationic formulation. So here we have our lipids in the solvent, again, aqueous phase. We make the liposomes and then we surface absorb the antigen. And the idea is you could have this going on in parallel, so your antigen can be prepared and your liposome simultaneously to save time. So here's, again, we looked at the various parameters. We looked at the flow rate ratio and the total flow rate, and you can see the similar trend that the 3:1 bringing that size down and 5:1 bringing it smaller. Generally, the 3:1 is the one we would go for with a smaller size, as we've seen before. But across the spectrum, you can see all flow rates and flow rate ratios, we get this lovely cationic nature, which we want to maintain. Because they're all nice and cationic, we get nice protein loading. So this is H56, the antigen for tuberculosis. Good loading, around 90%, as we would expect. But another thing we were very keen to check was lipid recovery, because these two lipids have very different solubilities, and we didn't want to lose one or have precipitation in the cartridge. And that was a risk. So we can see here we monitored lipid recovery, and it's good for both DDA and TDB. So we get both, and we keep that 5:1 weight ratio, which is very important. So that was great. So the next thing was, how do they form after injection? Because we know biodistribution is turning out to be a really interesting correlate of performance for us. So here we made the liposome. So CAF01 lipid hydration, so that's a standard one, and then our microfluidics at 3 different flow rate ratios. So then very different sizes, as you remember. They were 800, 200, and down to 100. You can see that. So this is the liposomes moving away from the injection site, and what we've actually also included is the area under the curve. So you can see they're all around 11,000 to 13,000% dose per hour. And there's no significant difference in how these formulations act at the injection site. So the liposomes are all sitting there. It doesn't matter what size they are with this formulation. They all hold their antigen at the injection site similarly again. So the bottom graph is now us tracking where the antigen's going. It's sitting there with the liposomes and will be, hopefully, we assume, co-presented to give us that delivery and immunogenicity. So again, with these formulations, with the cationics, we're seeing the size is not impacting on how they are moving within the injection site. So we also wanted to obviously check how they worked as in efficacy. So we did a standard vaccine model, we 3 vaccinations, and we monitor immune response. So the light grey is our standard lipid hydration that we've been using, the non-scalable. And the darker grey is when we use the microfluidics platform, so the nanoassembler. So in terms of antibody responses, equivalent, so that was great. So this is IgG2C. With IgG1, again, equivalent responses. So we can move from a small-scale batch to a effectively scalable or continuous process with no impact on antibodies. But obviously, as I've said before, cytokines are more important for us. So we actually looked at our cytokine production, in particular interferon gamma, which is our correlate. So again, you can see the lipid hydration versus microfluidics. Equivalent responses shown as that we now have a scalable or pop-up production method, giving us both that in terms of at the splenocytes and at the draining lymph nodes. So that's great. We were able to move away from this batch production nicely. But also what we've learned is really, for us, for these formulations, all the action is at the injection site. So whatever we do to make our cationic formulations move to the lymph node was not improving immune response. So for us, we were thinking, okay, let's not bother moving it to the lymph node, just let's make the injection site where all the action happens. Let's really increase the immune response there. So we looked at adding in pattern recognition receptors, so TLR receptors, agonists. So we looked at using resiquimod, so it's a small water-soluble agonist, and it's TLR7. If you use it on its own, it rapidly dissociates from the injection site because it's a small water-soluble molecule. So the idea is to conjugate it onto the liposome, and there has been some other work looking at conjugation of this molecule onto particles. So we took our DDA-TDB formulation, and we basically conjugated on resiquimod. This was done in collaboration with Eric Lapman, again at Aston University. So we take our DSPE lipid, and we do a two-step conjugation to get our lipid with the TLR agonist absorbed onto it. So now we basically have our DDA, we get TDB, where antigen absorbed and then conjugated onto the surface as a TLR7/8 resiquimod agonist. So what you can see in the graph here is the first formulation, DDA-TDB, H56, is a normal one. All the antigens stuck to the surface. If you look at the second bars, you've got the liposomes with the resiquimod conjugated. So you've got antigen there and the agonist, resiquimod. And the third set, you can see we just took our liposomes and mixed them with resiquimod. We wouldn't expect it to stay with the liposomes, and sure enough it doesn't because it's water-soluble. Agonist, it doesn't stick to the liposomes. So you really need the conjugation to get the two together. And as you would expect, if it's not conjugated, it just moves away from the liposomes very quickly. It will wash away. Whereas the conjugate stays with it. And the conjugate doesn't impact on how well we can load the antigen, which was a consideration. So we've got a liposome, we've got a DDA, we've got a TDB, we've got our antigen stuck to it, and our agonist now. So how does this work in terms of biodistribution? So here we can see over 1 day, 4 days, and 8 days, our normal liposome profile, DDA-TDB, you can see the liposome sitting in the injection site, and it doesn't matter if they've been mixed with resiquimod or the resiquimod's conjugated, the same profile. So the liposomes move the same whether the resiquimod's conjugated or not. In terms of the agonist, only when it's conjugated to the liposome does it stay at the injection site, as you would expect. If you just mix it with the liposomes, the resiquimod flows away. But when you conjugate it, the cations sit there holding the resiquimod, so they've got that. What happens when they make it to the lymph node? Again, doesn't matter if the resiquimod's conjugated, the liposomes move to the injection to the lymph node. However, only those with the agonist conjugated take the agonist to them. So you can see DDA, TDB with conjugated resiquimod all end up at the lymph node at low levels, admittedly, whereas when it's just mixed with it, it goes there and is cleared away quickly. So that conjugation really helps get everything from the injection site to the lymph node. So how does that look in terms of immune response? So our light grey bar is our DDA, TDB with the resiquimod all mixed together. Our black bar is when it's conjugated to the surface, and our light grey bar, the third one, is just a standard benchmark, no resiquimod. So you can see the black bar is generally higher. So when we have DDA, TDB, conjugated resiquimod, we're getting higher interferon gamma responses, which is the correlate we really look for in terms of that response, both at the spleen site and the lymph node. So this additional conjugation can boost the response further for us. So that's great to see. So we can actually manipulate the formulation more by considering what happens at the injection site, because we can't move the formulation to the lymph node without impacting on its immune response. So in summary, what we've now been able to do is we can do scale-independent production of liposomes encapsulating an antigen, all those where we have surface absorption. We can manipulate the formulation to produce a depot or to move it away from the injection site. We can cause antigen-presenting cell recruitment, and that's dictated by the formulation, how well they come to the injection site or how well things move. And we can make things move to the lymph node if we want to, and we can trap them at the lymph node using a conjugation system. So we have the ability to manipulate formulation, move the liposomes where we want them to go, and to take their antigen with them. So basically now we've got a fast, cost-effective, reliable manufacture, and we can drive these systems to give us strong immune responses. And just to say, as Jane alluded to at the very start, we are an innovation hub here, and people are very welcome to come to Strathclyde, and we will support you and help you get some hands-on experience with the equipment so you can test it out before considering purchasing it. So we have various of the systems here available in the lab should you want to come and give it a go. And to acknowledge this is obviously a large team we have working on it. So we're working on both vaccines and drug delivery, and we work in collaboration with Dennis Christiansen at Stanton Serum Institute, the guys at Aston, Andrew and Eric, and we have the team here in SIBS, Craig, Gavin and Simon, and also Nicholas Zittler down at UCL, who is also a strong collaborator. And thank you. One more. Open for questions.
Thank you very much, Yvonne. Do we have any questions in the audience first?
Hey.
Really interesting. I guess I'm a little surprised that you're getting better immune responses when you have localized at the site of injection. Whereas I would predict that you'd have to have it trafficking the lymph node and stuck on the follicular dendritic cells to get the strong antigen response. Do you think that there's this continually dendritic cells that are sampling at the site of injection and taking it to the lymph node to drive these kind of these immune responses? Is that what you're thinking is happening?
Yeah, I think you're exactly right, because we do see small residual amounts, and there's a team in Geneva that we also work with, and they look for double fluorescence, and they do see a very small amount of double positive antigen presenting cells at the lymph node, and that's enough to drive a strong response. We have also injected DDA TDB directly into the lymph nodes, and we get a massive immune response. So I don't think the depot drives the immune response. We get a depot because it's cationic, and the cationic gives us a good response. I don't think, if we could move it without getting rid of the cationic, we would, but we can't figure out a way to move these cationics without hiding the charge, which then downregulates their immune response. So I think they're two independent events. I don't think they're correlated.
Thank you, Justin. Any other questions?
Okay. Thank you, Yvonne, for another great presentation. I have a couple of questions, if I may. Do you have any idea about, you said about the carbon chain length that drives, do you have any idea about the packing that comes with it?
Yeah, that's, not really at the moment. We haven't looked at that in detail. So we've done some cryo-TEM, and the liposomes just look normal, but we haven't done any like Langmuir trough in that respect to get any in-depth detail. And we don't know whether maybe there's some impact on solubility of the different lipids in the solvent. So there's some of the guys are working on that at the moment to see, is the size more related to how soluble the lipids are in the solvent, or is it to do with the actual lipid packaging?
And also when you said about the ratio of the SPC to cholesterol, because that will drive also the rigidity of the
Exactly.
Do you have any idea? Because I think you did try to use the extrusion, right? Did you find any more resistance when you changed?
They're also large. I mean, we're trying to bust down something
non-scientific.
from. Yeah.
I have just another question. Use the DDAB cationic lipid. Is there any? But you didn't mention. Is there any difference between that one in DOTAP and DODAP and other?
Yeah. So we have looked at that. So we did a trial where we took DDA TDB and then DOTAP TDB DC-CO, TDB. I think those are the only 3 we've tested. Oh, and the DODA, so the lower one. So definitely the lower fluidity didn't work so well. DOTAP, not so well in DC-CO. So when we screened against those 4, they were, DOTAP always, DDA always comes out as the highest one. Well, I mean, we haven't exhaustively tested them all, but yeah, we have looked at. I mean, the other one we did look at briefly was stearolamine, which is basically half a DDA. We tried that many years ago, and we didn't find it as good. So, and it doesn't really form liposomes as well, so that's the thing. So,
did you see any change in the charge of the ones that you could form?
From? No, I think not really at the ratios we were working at, because they're very cationic. We might be able to tease it out if we played about with buffers, we might be able, but generally they're all above 40 millivolts because we're putting like so high amount in.
Okay, thank you.
Do we have any more questions in the audience? So we can take some questions from Twitter now. So this is in relation to the TFF. So could you comment on the dose concentrations in weight per volume achieved by TFF nanoparticle concentration? And do you have any aggregation concerns with this?
Okay, well, you could probably answer it as much as I could, but, no, we haven't seen any problems with aggregation, as you know. So in terms of concentrating up, I think we've got to about 40 megs per mil, I think. 60, okay, so Carla's got to 60, I think you got similar, didn't you? Yeah, so we have got quite high, and we can concentrate it up quite nicely. I think that's, we've had no problems, have we? No.
Yeah. To that point as well, so for my project, one of my formulations was a very anionic formulation. So we had quite low encapsulation efficiency. So instead of just pumping it full with protein and getting really poor, inefficient loading, we would scale it down a bit, and then we were concentrating with the TFF.
Yeah, so that's
and
good, yeah.
we saw no issues with aggregation.
Yeah,
but these were anionic particles, so we did have a charge repulsion going on. So
yeah, and that is a good point. Yeah, we've never managed to be able to do TFF with cationics. None of us have we? We lose it in the TFF column. I think we've all had that beautiful experience of no liposomes coming out, or they change charge. So I think even our guys that are working in other labs have seen similar, so we see that drop in zeta potential. And we fluorescently label them, and our column goes pink. So, yeah. So neutral, negative, great. Cationic, we're still trying to fix it.
Thank you very much. So we have one final question here. So how do these DDA, TDB, resiquimod conjugates compare against human vaccines adjuvants, such as aluminium hydroxide gel or others that are known to cause a depot effect?
Yeah, that's a good question. So the aluminium hydroxide is not brilliant for TB because we want more cytokine interferon gamma. So yes, it's much better. So the early papers by Staten Serum Institute did the benchmarking for us again. Well, they did the benchmarking on their own, and they have shown that their liposome formulation, because they did develop DDA, TDB, is much stronger in giving the immune response profile that they need compared to alum. In terms of if you were going strictly antibody, I don't know what the literature is. You probably get a fairly reasonable one, but certainly in the cytokine and interferon gamma, DDA, TDB is given as much stronger response. I know there's controversy also against the depot in Freud's adjuvant and aluminium hydroxide. They do cause depots, and again, is the depot the driving force? Well, we don't really know. So, but definitely in that.
Thank you very much, Yvonne. So, if you have any more Twitter questions coming in, we can try and answer them just through our page ourselves, but I think that's us done for now.
Yeah, thanks.